The topic of this presentation is entitled Current and Future Trends in Preimplantation Genetic Testing. And the central question that is going to be involved is, can concurrent or current and newer methods designed to obtain detailed genetic information about embryo normality and developmental competence significantly improve outcome without raising attendant problems of actionable interpretation? What is actionable interpretation? Is how do we use this information to decide which embryo should be transferred, cryopreserved, or discarded? And this has been a central question since the onset of genetic testing for human embryos in IVF. And in some sense, it's gotten more complex, but in other sense, it has gotten much more apparent as far as what is actionable and what is not. And I want to start a little bit about the history of PGD and why it was, how it's evolved, and what concerns led to its use in IVF. And one thing we've known is it's long been recognized that aneuploidy is a likely cause of significant number of spontaneous abortions. And this has been known for quite some time, even well before IVF. And this has been the rationale, in fact, for prenatal genetic screening first by amniocentesis and later chorionic manila samplings that first used conventional cytogenetics, karyotyping with what's called G-banding or Giemsa staining, of metaphase chromosomes to detect aneuploidy, translocations, and relatively large deletions in individual chromosomes that can be observed by light microscopy. Now, this method was first used to examine meiotic metaphase chromosomes in residual IVF and GIFT oocytes, GIFT being gamete intrafallopian tube transfer, which during the early 80s was actually much more successful in outcomes with respect to IVF. And in many cases, since many oocytes were obtained, patients did not want IVF to be performed because it was an added expense. And actually, the results from IVF during this period were pretty poor, with outcomes of 10% seen in many programs, 20% in others, and even a little bit higher in some others which more experienced people. But it wasn't the level of success rates that we see today, or hopefully, as you'll see from this talk, in the future. But what was evident from just looking at oocytes at this time was that high rates of aneuploidy did occur in the oocyte prior to fertilization. And in some reports that were published in during this era, the aneuploidy rate was 50% or greater, which is an astounding number. And later, as you'll see, FISH, fluorescent in situ hybridization methodology, was introduced to use specific DNA probes coupled with fluorophores to test for numerical chromosome analysis and use of single blastomeres physically extracted from an 8-cell embryo. So you had a change from FISH, from karyotyping by light microscopy to fluorescent microscopy using specific probes to specific regions of DNA, and a single blastomere usually extracted from an 8-cell embryo. Aneuploidy findings were assumed to be representative of the chromosomal status of the embryo at large for purposes of transfer or discard. So if you had an embryo biopsy from an 8-cell that was normal, chromosomally in terms of chromosomal numbers, then that was assumed to represent the entire embryo, whereas if one was aneuploid, a monosomy or trisomy, that actually was a signal that perhaps this embryo should not be replaced. And there were also concerns expressed about damage to developmental competence by retrieving one or more blastomeres. And to show you a little bit about this, the early karyotypes that were done before FISH were similar to what's shown here. And in those days, what you'd have is a photograph that you took from a light microscope, and that you would hope that the metaphase preparations allowed you to see each chromosome. This is a G staining one where you could see banding patterns, and then you'd have to cut out the individual chromosomes and realign them manually on a sheet of paper to get a karyotype of this sort. And in fact, if you look at this little bit, you can see some of the outlines of the photograph that was taken. Meiotic chromosomes were a little bit more difficult to image, primarily because they're more condensed, and there was more overlap that took place, which made it a laborious process. However, instrumentation came about for amniocentesis, primarily, that automatically took pictures from slides, and you could do multiple cells in that case, and then would rearrange using a cursor each chromosome, and then would align them 1, 2, 3, 4, 5, etc., depending upon the chromosome number, automatically. And this was a huge labor-saving device, and you could do multiple cells, because if one cell from an amniocentesis was abnormal, you had others to look at the same time, so you could assess the relative abundance of normal and abnormal cells. And this was a very big improvement, because it allowed oocyte and early embryo biopsies to obtain chromosomes both from meiotic and mitotic cells for this purpose. But again, it was laborious and required a lot of effort to do this, even with the instrumentation. However, a number of interesting findings were derived from this, and that is, if we looked at the larger numbers of oocytes, this was a study that we published in '92, and there was a previous one published, I think, in '87 or '88 in Human Reproduction. And what was found by looking at specific groups of chromosomes, where you knew what the frequency of aneuploidy was in abortions, which is shown in the first line, where you'd have, let's say, percents of chromosome from A, B, C, D, E, F, and G groups, which represented groups of chromosomes based on their size, that in fact you could compare those to what was occurring in the oocyte. And so when you did this sort of analysis, what was found was that group C chromosomes, for example, were actually lower in aneuploidy in oocytes than they were found in abortions, but for D and for G, and especially in G being the small chromosomes like 21 and 20 and 17, that in fact the incidence of aneuploidy, both trisomy and monosomies, were higher, a lot higher than you would see in abortions, which means that in fact a high percent, when you add all these figures up, of oocytes that were obtained for this type of analysis that would be used for either, in this case, GIFT or IVF, had a higher likelihood of being aneuploid than not. And this was important because it confirmed what a lot of people were seeing at this time, that aneuploidy was a significant issue. And it was sort of difficult to undetect, except in embryos that failed to progress in culture. You could see them stopping at the 4-cell or 8-cell stage, and you'd say, yes, these have maybe aneuploid. But in embryos that progressed and looked normal, in those days most embryos were put back on day 3, that it was hard to tell whether or not an embryo was aneuploid or not. And so this was a difficulty, and there was some risk, and so this led to two things. One is to put back large numbers of embryos, in the sense that some of those that were aneuploid would not develop, especially in the lower groups A, B, C, and D, because you really never would see these as much in aborted fetuses. But there was always the risk of, for example, trisomy 21. So a lot of those trisomy 21 embryos would really not implant or not progress, or would not progress through the early stages of development after implantation. So there was some risk, but it was, I guess, at the time, a reasonable risk. And so more embryos were put back than we do today. And that resulted in findings that were a little bit disconcerting, because you'd have pregnancies that were higher order gestations, quadruplets, quintuplets sometimes, and triplets and twins. And this was not a good outcome for the patients, maybe for the clinic because it was good advertising that they were successful, but not necessarily for the patients, especially with the much higher order gestations. So some other method needed to be done to be able to sort of assess without really a damage to the embryo the frequency of aneuploidy. And this came out with fish. So we went from the old school karyotyping, which I just showed, to now what is called the fluorescence in situ hybridization to detect specific gene sequences on chromosomes. And this was very useful because you could take one blastomere and actually find the regions, chromosomes that were present in abundance or in deficit, the aneuploidy. And this was used for a while until another method came out, and that was multicolor fish. And this was a spectral imaging that allowed you to look at all the chromosomes because each one was a different color. And by doing this, you could look specifically at aneuploidy from a blastomere. And this was very, very useful. Not easy to do and very highly specialized, in fact, to do it. You needed equipment or commercial labs were doing this sort of analysis. And here you can see an example where there was a missing Y chromosome. So you have X, X, O, or XO in this case. And this allowed you to look at certain types of defects that were occurring, including Down syndrome, Klinefelters, Turners, etc. And this was used for a while to look at karyotyping in this way and to determine whether or not an embryo was normal or not at this level of, with respect to ploidy. And this was used, as I said, for quite some time until another method came around. And this is going from this MFISH to competitive genomic hybridization for aneuploidy detection. And here is kind of interesting because in this case, what you have is a way of looking at the entire genome. And this was not developed for IVF purposes. It was developed basically for molecular genetic research, where you would look at hundreds, if not thousands, of specific genes and try to determine in a certain type of array of these genes manufactured by a company, for example, whether a specific gene or a set of genes or a family of genes was overexpressed, underexpressed, or normal. And this was used both clinically, for example, in trying to assess tumor heterogeneity, as well as in research studies of looking at essentially gene expression during development in a variety of systems. So companies started making these arrays, and they were very, very useful, but you'd have to have the equipment to see it. So competitive genetic hybridization is used to quickly scan an entire genome for chromosomal ploidy and genetic imbalances, deletions and mutations, for example, and does not need cells that are undergoing cell division. And that's an important aspect of this, because with fish, you really do need cells at a specific stage and presumably, hopefully in metaphase to determine the best signal. And the primary advantage of competitive genomic hybridization is the ability to simultaneously detect aneuploidies, deletions, duplications, and or amplifications at any locus represented in the array, with one assay being equivalent to thousands of fish analyses. And I have a recommended reading that's going to be in your list that you might want to look at to talk about the methodology as it developed of competitive genomic hybridization. And to give an example of this, shown here is the type of a portion of an array that you would see shown on the right-hand side, upper right-hand side. And these are commercial DNA microarrays that contain thousands of genes in individual spots deposited on a membrane or a glass substrate and then put into a machine after you've added your sample that you prepared from an extraction of the DNA or a whole cell, and you put this in the array, and you have microarrays also for specific gene sets that might be of interest for metabolism, mitochondria, specific diseases, signaling pathways, etc. And the machine looks at these and gives you a printout of each of these genes and from each of these spots in terms of their relative abundance, normal, high, or low. And the next sequence, since that was done commercially and was fairly expensive actually, was done for a while until another technology came around which was really important, and that's next-gen DNA sequencing. And if you think about DNA sequencing that was done for the Human Genome Project, required a lot of effort and machines and doing sequence. It was a long-term laborious process. Next-gen sequencing changed that all with instrumentation that allowed you to do rapid, high-throughput sequencing of DNA. So here we've gone from, for IVF, from competitive genomic hybridization to next-gen sequencing. And that resulted in a family of pre-implantation genetic testings that were available. PGTD was originally described as pre-implantation genetic diagnosis, which was changed to a more aptly termed pre-implantation genetic testing, because you're not doing a diagnosis, which would imply that you're looking at a specific gene, but rather a screening for ploidy. And these tests were PG and PGTM, which can test for a specific monogenetic disease, such as cystic fibrosis, Duchenne's muscular dystrophy, Fragile X, Huntington's chorea, etc., as noted here. PGDP, which is pre-implantation genetic testing for polygenic disorders, by estimating the risk of developing adult-onset diseases that are multigenetic or multifactorial by examining a combination of specific gene variants. In other words, a lot of diseases are not single gene, they're multi-gene, and this allows you to test for the presence of those multigenetic variants that would result in a disease state. PGDA is the most common used in pre-implantation genetic screening now, and it's used to detect aneuploidies such as Downs, Klinefelters, Turners, etc., with more recent methods identifying structural DNA defects, which is very important, such as microdeletions, duplications, inversions, which occur on specific regions of chromosomes. It's also used, by the way, for male in testing for Y-specific or Y-located deletions, duplications, etc., mostly microdeletions. And there have been many microdeletions of relatively short sequences of DNA on the Y chromosome that have been clearly associated with infertility, including azoospermia, reduced sperm production, oligospermia, and problems in the actual formation of the sperm during its spermatogenesis. So this is actually important for testing and used for testing men for these microdeletions, including what's called the most prominent one, the DAS mutation, which is deletion azoospermia. So PGTA is what we use. Now there's another variant which is not often discussed, which is called PGTSR, which structure incorporates into the PGTA for structural abnormalities, as I've mentioned, which are called microinversions, deletions, duplications, and translocations, which may or may not be important, as you'll see later on in an outcome, but soon to be including epigenetic modifications, including methylation, which are actually more important than some others in terms of the normality of DNA function at specific stages of development. So these are the families, with PGDTA being the most important. NGS involves parallel sequencing of multiple small DNA fragments recombined to determine an entire genetic sequence, genomic sequence. And the benefit is its high throughput significantly increases the speed at which an individual genome is sequenced and displayed for analytical purposes. Now recently, artificial intelligence has been applied as an enhancement that has the potential to markedly increase genetic information content because it detects and prioritizes genomic variants linked to certain diseases that are obtained from large databases using large population of normal and affected individuals. So here you have not just the sequence, but artificial intelligence algorithms included that as an enhancement is able to more accurately pick up and detect abnormalities and that lead to problems from the entire DNA sequencing, not just ploidy. And so this is something that has been introduced and actually may be very, very important to increase the sensitivity of these assays for embryo selection. However, all of these karyotypes and FISH and competitive genomic hybridization and next-gen sequencing confirm what has long been known, and that is that the frequency of chromosomal aneuploidies, and likely also probably structural defects, increases with maternal age. And as it increases with maternal age, the viability of the embryos decline coincidentally. So this has been sort of an important verification of, or validation of, what has been known for a very long time. And it's been used, of course, in the basis of pre-implantation genetic testing. Even with the enhancements, it's even gotten a little bit better, such as artificial intelligence. However, all of these procedures, or most of them I should say, have a number of problems associated with it that have been sort of complex problems. Mosaicism has been described as one of the most important ones, and mosaicism is seen as early as in FISH analyses were introduced years ago, and is very common in NGS reports. So what is mosaicism, and what is the relevance? Because as with aneuploidy, it is not an uncommon finding. In many reports we get back from doing next-gen sequencing of DNA now includes both a mosaic component to it. And how do you use that information? Well, from FISH, it was clear that a healthy embryo has 2 chromosomes. An embryo, for example, with Down syndrome, from a single blastomere assay, it's the early days of FISH, has 3. And an embryo that has, for example, Klinefelter system, is like XXY, has 2 X's and a Y. And you could see that. And that was in a single cell. And you could say, well, gee, I wonder if it's in all the cells, and what do you do with an embryo like this? Do you put it back or not put it back? And most of the time, embryos that were obviously with Down syndrome or other chromosomal triploides or monosomies, trisomies or monosomies, were not replaced. I mean, that's the whole basis of doing the assay. And the same is true where you had mosaicism. You could say, well, it's in one cell, but is it in all? And that was more difficult to understand. But again, if you didn't have a healthy embryo with a normal karyotype, likely those embryos were either not cryopreserved or were not replaced. And here's an example of what you'd see for current next-gen sequencing report. At the top is a normal profile that you would see. At the bottom, in the middle, is one that had mosaicism, and that mosaicism could occur in multiple places. And all these are trophoblast biopsies, so you have multiple cells, not just one. And in these cases, you have the question of whether this mosaicism is relevant or not. And if you have multiple mosaicism sites that you're seeing in these different chromosomes, here's a mosaicism in one particular chromosome, but often it comes back with several chromosomes that show this type of pattern, as opposed to the one at the bottom, which is aneuploidy, and that's pretty clear cut. And usually you can have aneuploidy at several chromosomes where you have an abundance below and above the normal line indicating the absence of a chromosome or its presence in excess. So that's clear cut. In the normal, it's pretty clear cut in the case of the aneuploid, but it's not so clear cut in the case of the mosaics, and this has always been the problem. So a lot of work has been done in this regard, a lot of work, and that work has been published and has led to a sort of interesting findings, including, as you'll see in your reference list, a consensus of opinion about what to do with mosaicisms. And that's a very recent report. And the question is, does more detailed genomic PGT findings create more confounding issues such as mosaicism for transfer, cryopreservation, or to discard in the decision-making process? And one consensus is that some proportion of cells in a preimplantation embryo screened for BGTA are aneuploid. That's pretty clear. But what is the level of aneuploidy? And when you're dealing with multiple cells used in the process of screening for next-gen sequencing, it's been suggested that pretty typically about 20% or less, mostly less, of aneuploid cells, their occurrence is considered normal. So this is not a concern if you have an embryo that comes back like this, with this screening. With an aneuploidy rate of, let's say, 10 or 15%, it's considered normal, and that's not going to affect its or compromise its subsequent developmental ability and normality. It gets a little bit more complex when you get 20 to 40% level. And although that's considered low, some programs will not replace, others will replace, with the provision that you may want to do what is now used routinely and is maternal blood screening for fetal cells, fetal DNA. And this is now sort of supplanted amniocentesis in most cases, and certainly chorionic villus sampling. So you can pick up DNA that is shed by the placental cells, and that DNA is representative of the developing fetus. So if there's a concern, you could follow this up with the maternal blood analysis for fetal DNA as a reassurance that things are normal or not. When you get to the 40 to 80% level, that's considered high. And in that case, it's likely, and especially with greater than 80% where the embryo is considered abnormal, that in fact you probably don't want to put these embryos back. Although again, it depends upon the program, and a lot of times there may be some consideration for doing that, but most of the time in our program and others, that an embryo like this probably would not be returned. It might be cryopreserved, but certainly not returned in a fresh cycle. So often, the detection by next-gen sequencing for PGTA reports of structural anomalies, microdeletions, or mosaicism has been used to err on the side of caution to preclude transfer or cryopreservation. So that's been the sort of the history, as I just mentioned, that you always have in some programs when a report comes back which says, well, there's mosaicism or there's some structural defects, that can't be good. So structural defects being the inversions or microdeletions, etc., that's seen in one or more chromosomes. And so those are not returned. They may have been cryopreserved, but not returned unless completely normal, a normal euploid embryo without significant structural defects or mosaicism levels. But there have been some widely discussed, published studies and comments in the popular press from very prominent members in the IVF field, suggesting that, according to this dogma, that or protocol, that thousands of embryos may have been needlessly discarded because recent outcomes indicate that such embryos can result in normal babies. And that calls into question a lot of things, okay? The relevance of this testing to begin with, but the relevance of these sort of abnormal, what would be considered abnormal findings. And this has created clinical and patient confusion about whether and which PGT findings are truly actionable. In other words, what do you use them for? And in some notable instances, at least we have seen in the United States, patient-initiated litigations have occurred that involve IVF programs that failed to transfer embryos when all were suspected based on PGD results, or were suspect, in other words, based on PGD results. So, what is the value of PGD to begin with? And that has recently come into question as well. And one study, among many that are like this, by Yan et al. In New England Journal of Medicine. And their central question was for trials evaluating PGTA, this is how they did this, a cumulative live birth rate reflects the possible effects of the practice of discarding some embryos that might have led to a live birth if they had been implanted. Such data cannot be captured when only the first embryo transfer is analyzed. So you have lots of embryos, you put one back, and it didn't work. Okay? So I think if that one didn't work, which was supposed to be euploid, then this could be a problem with the rest of them. And in some programs, they were reluctant to do even frozen embryo transfers on similar embryos. So their study design was as follows. They compared cumulative live birth rates after PGTA based on a combination of morphological criteria or next generation sequencing with the rates after conventional IVF based on morphological criteria alone. So here you'd have some embryologists who would look at these embryos and say, yeah, it looks pretty good to me, and we'll put it back and send it out for next generation sequencing. Let's see, blasts and trophoblasts, cells from blastocyst stage embryos. And the other one is just going to look at the morphology, no next generation sequencing. So one, you have morphology and next generation sequencing, and in the other group you have good morphology alone, that using criteria that are pretty typically used for assessing blastocyst normality. So in the PGAT group, euploid blastocyst was chosen for transfer. In the conventional IVF group, a blastocyst was chosen based on morphological criteria alone. If live birth was not achieved after the initial transfer and there were remaining euploid embryos in the PGA group or the morphologically transferred group, subsequent single embryo transfers were performed. And what did they conclude? They found that among women with 3 or more morphologically good quality embryos, conventional IVF without any genetic testing resulted in a cumulative live birth rate after 1 or 2 transfers that was non-inferior to the rate with PGTA, which suggested that in fact the outcome rates were the same. Now you could argue that the difference was they had to use frozen embryo transfers. And so the argument in this case is that the TTP, or time to pregnancy, may be better if you include the next generation sequencing than morphology alone. So it remains to be determined what people want to do. Some programs like to do PGT testing on all embryos, some on only some in patients at risk. Others prefer to do, for example, morphological screening based on developmentally established landmarks. I'll talk about that later. In terms of, for example, when pronuclear formation occurs, when pronuclear dissolution occurs, when the speed of compaction, the speed of cell division, and how the blastocyst looks at the blastocyst stage. And a lot of this is done by time lapse in instruments like the embryoscope and others, where you can actually monitor and record the progressive development of the embryos over a long period of time. And that can be used for embryo selection in terms of without any ploidy analyses. So it remains to be determined which one will work, but there may be other better methods that I'll talk about that can be applied. So now we have something that's really quite interesting, and that's the introduction of non-invasive pre-implantation genetic testing. What that means is you're not going to go in and pull out a blastomere like we had to do at the 8-cell stage, or you're not going to have to do a biopsy at the trophectoderm at the blastocyst stage for analysis. Yet you still can get important information about ploidy and other concerns that may be actionable information to decide about transfer or not. And so there are a couple of current ones I'll talk about. One is the analysis of static and time-lapse imaging during culture in a machine, in the embryoscope, for example, with or without the addition of artificial intelligence, which is a really important aspect of this, because artificial intelligence is a collection of large amounts of data from large amounts of embryos and patients that provides an additional information content that can be used to determine what works and what doesn't work in terms of morphology and in terms of, for example, the aspects of embryogenesis that you've looked morphokinetic and morphological during the period of time in culture. So you have now a data set separate from what you've seen that can be used as an assist for selection purposes. More recently, there's a collection of DNA from spent culture media after 5 to 7 days of embryo culture. And that DNA is then used for next-gen sequencing, like you would use for trophoblast biopsy, for example. And then there's the manual collection of blastocelic fluid. And the manual collection of blastocelic fluid is a relatively recent application where you have to actually manually express the blastocelic fluid, collect it, and then or extract it from an embryo blastocyst physically with a pipette, and then that has DNA in it many times that can be used for genetic analysis. And of course, embryos expand and collapse in culture. We've seen this many times, that they undergo these periodic expansion and collapse phase at the blastocyst stage. Some people believe that's an in vitro artifact, but they do collapse and then recover their blastocelic fluid relatively rapidly. And so, in fact, it's not a detrimental procedure to collect it either by manually collapsing the embryo or extracting the fluid. And in fact, it's been suggested that collapsing the embryo prior to vitrification may in fact improve outcome, because it is more effective in the incorporation of the cryoprotectants and their release later on during thawing than it would be if the embryo was attacked. And with vitrification, the embryos often collapse, blastocysts often collapse anyway. So it's not necessarily a toxic manipulation that is occurring, and you can get blastocelic fluid. So the introduction of artificial intelligence in all of these systems, as you'll see, these are artificial intelligence algorithms in non-invasive embryo selection is another recent enhancement that has been placed on both next-gen sequencing and on the morphological only determinants of development in vitro, for assessing embryo competence, normality, and potential outcome. So one question is, can the inclusion of AI algorithms in PGTA significantly increase the accuracy of aneuploidy and euploid identifications, perhaps even structural defects, but I don't think so, to better select embryos suitable for transfer at the blastocyst stage? But you need evidence-based outcomes to prove a significant increase in ongoing pregnancy rates occur, as well as live birth rates, compared to static morphological or just gene DNA sequencing alone or in combination. So there are two recent reports from this year, actually, that are really quite useful in this regard. And they are detailed reports that suggest the use of their, as described in the papers, AI platforms and algorithms has a potential to achieve a very significant reach of these goals by outperforming embryologists' grading at the blastocyst stage. And this is what many of us do. They look at blastocysts, whether in one way or another, or even in an embryoscope, and look at the conditions of development, and make a selection of embryos based on those findings. So in this study, a retrospective study by Barnes, published this year, with over 10,000 blastocysts, used maternal age, morphokinetic, and morphological characteristic, typically applied for blastocyst grading, either static or in a embryoscope time-lapse culture system. And in this instance, static images at 110 hours post-ICSI. So all these are ICSI, by the way, experiments or procedures. These non-invasive findings were correlated with the corresponding PGTA results, that is from next-gen sequencing, and they showed a good predictive value, greater than 75% for aneuploidy, euploidy, and complex aneuploidies. It's pretty good. So it's simply based on these characteristics which are non-invasive, okay? And then they did the biopsy, and they correlated all this, that the concordance was greater than 75%. It's not 100%, but even with next-gen sequencing alone, you can often get embryos where there's no signal or no meaningful signal that was produced by the sequencing from that particular set of blastomeres, or if you do the 8-cell stage, or of next-gen sequencing from the trophoblast. So these early findings show promise for ploidy and may especially be useful if inner cell mass characteristics, now known to be associated with good developmental competence and normality, can be applied in the AI algorithm, either alone for next-gen sequencing or without or with time-lapse analysis that's been really prevalent now in the field. So these are really important findings, especially for AI, because AI is based on very large databases of outcomes of specific embryos. So this is very, very useful in that regard. And there's another highly recommended and excellent review by Yang and Borman again this year. And this discusses the current state of artificial intelligence in the application of embryo ploidy grading and its limitations, including what I described before, the issue of mosaicism, and also the detection of structural defects and the use of image-based analysis and DNA source. So in other words, where is the DNA coming from? Whether you're using it just on next-gen sequencing alone or in combination with image-based analysis or just image-based analysis. And their conclusion is that even with possible confounding factors, artificial intelligence-based ploidy prediction has the potential to improve pregnancy rates and, more importantly, reduce the cost of IVF cycles. So this was an important finding which you should read this study, because it says, yeah, there's a real place for artificial intelligence information to supplement and enhance the signals that you can obtain either from morphology alone, such as you do with time-lapse, or with next-gen sequencing alone that might be wanting, because you have this database to look at. However, not all in the field are sanguine about this. In other words, not optimistic that AI and image-based analysis is useful owing to the question of whether undetected mosaicisms and structural defects exist, or when detected may compromise actionable decisions for transfer. This is especially true if the affected embryos can result in normal outcomes. So there's some confusion that says, well, maybe all of this is not that definitive, and we still have some problems, so how do we deal with this? And a lot of people, myself included, feel if this is the case, that this would tend to argue for a gold standard finding obtained by NGS, because you're looking at the entire genome. So there are current considerations for non-invasive ploidy detection, and they call it cost-benefit. So one of the problems that could be an issue is the likely need for ICSI, because you want to look from the very beginning, especially if you're doing morphological assessments. If you're just letting them grow and culture and doing static imaging by embryologists at specific stages where you think the important landmarks of development are occurring that have to do with speed of first cell division, the timing and morphology of pronuclear dissolution and formation, when compaction occurs, when blastocyst formation occurs, and what the blastocyst looks like, then in fact AI may be quite useful in that regard. So you have ICSI, you need probably multiple time-lapse culture systems to accommodate embryo numbers to be examined, especially in very active IVF programs. And you have the cost of non-public AI algorithms if not included in the culture system itself. So where do you get these algorithms from? Some may be sold and some may be freely available in public domain, as in the two papers, by the way, that I just previously referenced. Those are publicly available, what they use and can be used by you if you want to so use them. So it'll tell you which chromosomes are trisomy and monosomy, and that's very useful, whether translocations can occur. You not likely need IVF programs to do self-validation with coincident NGS-PGT at the blastocyst stage to make sure if they're using morphology and morphokinetics alone, that in fact the paradigms are working for them. And one solution that has been suggested many times is you reserve these types of analyses that are complex and provide a lot of information for couples considered at risk and where PGTA would be normally recommended. So this tells us that there are a lot of options that you have based on the type of information you're collecting, where it's collected, how it's analyzed, and how it's interpreted. However, there is another non-invasive method of PGTA that I just mentioned that has recently been introduced, and that's the use that uses cell-free DNA for genomic sequencing. Now, non-invasive PGTA using cell-free DNA is not new. Prenatal genetic analysis has evolved from invasive amniocentesis, as I mentioned, in CVS, to cell-free capture of DNA in maternal circulation shed by apoptotic placental cells. And similarly, DNA detected in spent culture medium is by definition non-invasive, whereas blastocytic fluid obtained from manually collapsed embryos is considered minimally invasive for PGTA purposes. And of course, the embryos usually will re-expand. Of course, collapse in culture is not uncommon. In validation studies, some reports describe an acceptable level of concordance with DNA extracted from corresponding trophoblast biopsies, while others are less so optimistic about that. But in general, there seems to be a pretty good correspondence from culture media, and to some extent from blastocytic fluid. When or why or how DNA occurs in culture medium or blastocytic fluid is unclear, but apoptosis is known to occur in the trophectoderm of several species at the expanded blastocyst stage. And when you do, for example, tunnel analysis of blastocysts, you see cells that are tunnel positive normally, and these are normal embryos. And in fact, in some species, like the bovine, it's quite extensive, and in rabbit, it's very extensive because the rabbit blastocyst may have nearly 1 million cells in it. And so an origin from the trophectoderm is not a surprise. It depends upon the amount of DNA that is released and whether that DNA, in fact, is analyzable and can be amplified. So a similar origin would seem likely for the human, especially for the blastocytic fluid. But it's said that embryo-derived DNA from culture media is, while truly non-invasive, and it seems from several studies that, as I said, the concordance with trophectoderm biopsies is in fact superior to that of DNA from blastocytic fluid. And that's an important finding. However, for both sources, significant PCR amplification is required for meaningful signal from DNA sequencing, so many of the same issues of concern with nuclear DNA for NGS from intact cells may apply. And this includes signal fidelity, amplification, etc., all sorts of problems that can occur with NGS sequencing depending upon the quality and amount of DNA, and problems with amplification of the DNA, how many rounds of amplification are required so as not to develop artifacts in the signal, the noise that may occur. For culture medium, robust denudation of cumulus and coronal cells is required, and it has to prevent maternal contamination. And for even from a handful of residual cells from coronal and granulosa cells are a problem, especially when you're doing culture medium or even blastocele. Well, it wouldn't be blastocele, but for culture medium. And this may require aggressive ICSI, as with conventional IVF, it can be difficult to assure complete maternal cell elimination. However, the relative benefits of cell-free DNA for non-invasive PGT remains to be settled, but is conceptually relevant in IVF as a new mode of assessing embryo genetic normality. As I said, in some reports, blastocytic fluid has been considered unacceptable for PGTA owing to high levels of amplification failure, poor fidelity, and or high discordant rates with coincident biopsied trophectoderm extracted DNA. Collapsing the blastocele prior to vitrification has been suggested to improve post-thaw viability in vitrification, as the blastocyst cavity should reaccumulate fluid, and it usually does. And usually during vitrification, the blastocyst cavities often collapse spontaneously and then re-expands on the process of thawing and culturing it. However, full re-expansion is not a given, and failure to expand or return to a pre-collapse state may indicate a reduced metabolic capacity. That is an inherent problem with that embryo, which in itself could serve as a further marker of developmental competence. So a lot of this needs to be resolved, and will be resolved, I think, in short order, and pretty soon. But again, you need additional validation studies. It would seem that currently collecting cell-free embryonic DNA in spent culture medium could be the most cost-effective means for PGTA, allowing the power of next-gen sequencing to determine ploidy, structural defects, translocations, and for monogenetic mutations, and as well PGTM. So I think there's a lot of possibilities here, and we'll have to see how that turns out. So I would say a couple of take-home messages. One, that many aneuploid embryos will self-deselect by arresting development prior to the expanded blastocyst stage, even with extended culture to day 7. So what you're looking at is a relatively unique set of embryos from a large cohort that you obtain by aggressive superovulation, essentially, in human. Get a lot of eggs, a lot of embryos. Some of those, some higher or a lower proportion, will stop dividing, and they will never be available for biopsy or for transfer because they deselect. It's the ones that keep going that have characteristics that may be indicative of normality, both at the ICF and trophectoderm level, obtained by just in operator inspection or by time lapse with the presence or absence of assistance with artificial intelligence data that you're concerned about. And I think one aspect of this that is still needs to be resolved is personal opinion on the part of the clinician. At present, if you believe genomic sequencing is the gold standard to detect genetic issues that may compromise outcome, even if morphologically stage appropriate for ICM and trophectoderm, then NGS is indicated. And it's invasive. Okay. You may want to do it by culture media, or by, probably most likely culture media, or by trophoblast biopsy. In this regard, DNA in spent culture media has the promise of avoiding invasive intervention and should be cost effective for the patient, and if proven to be equally accurate as cellular DNA, an alternative may exist to time-lapse imaging even with artificial intelligence. So I think that remains to be determined, but it's a direction in which this field is going into, and if positive, I think will be quite important. So what are the future prospects? And I'll end just with a couple of comments about where this is all going in the future, near future. And I would just...
My presentation concerns future
and that is modern cell biology
pre-implantation
has advanced tremendously in its ability to examine molecular, metabolic, and specific gene expression at the single cell level, often simultaneously in living stage. So this is currently huge changes in remarkable technology that has evolved in cell biology, molecular biology, and combining molecular biology and genetics,
important genetic information,
how cell structure and function works.
But there is a central question, and this is not a new one.
Combined with various omic platforms, insights into normal and abnormal cell functions can be assessed, and if applicable,
and that is
cells
current
extracted
and newer
from
methods
late-stage
designed to obtain detailed
embryos.
genetic information.
Similar
about
insights
embryo
into
normality
post-transform
and developmental
normality
competence
might be
significantly
estimated
improve
as a
outcome
final screen
without raising
for
attendant problems
for
of
developmental
actionable
competence.
interpretation. And
While
what I mean by
combining
that
PGT
is to
and
what extent does the
types of
information
embryo
that is
studies
obtained in
may be
greater
asking a
detail
lot One lesson
in
learned from
either
over 4 decades of clinical
clear,
IVF
actionable
is that the
results
imperative
about
to
what
improve
can
outcome relies both
transfer
on skilled
or
embryologists
create problems or
and not
doubts
go
about
away,
the
and the
relevance of
continual
information
development,
with
testing,
respect
and
to
application
outcome.
of
This is an
viable new
important aspect,
technologies
not only
that
in
can
genetic
clearly
testing in
provide
embryos,
different
but
streams
also in genetic
of
testing in
developmentally
general that is
relevant
done
information.
for clinical
In this
purposes, for
presentation,
example, in
the
assessing
evolution of
tumors
PGD from
and
chromosomal
other types of
karyotypes
illnesses.
to next generation sequencing,
And so the question
to the
I would
addition
start
of
with
artificial
is how
intelligence
PGD
is
began
a clear
and how it
example
has
of a
evolved in
progression that
clinical
serves
IVF.
this imperative.
And
And I think we can really
the question
anticipate
is really an
with all the
interesting one,
biology
because you
has
wouldn't ordinarily
offered
expect to
changes that have
have such
occurred
a
in our ability
tremendous
to
enterprise
look
that
at very,
currently
very
exists
precise
in
events
the
that occur in cells at the molecular,
testing of
cellular, and
embryos
genetic
in
level.
IVF
I think we
programs,
can anticipate that this will
being
continue to
mandatory, some
evolve
being,
and contribute to one of the
restricted
most
to certain
important
cases,
and
others
successful
not at
areas
all.
of medicine, that is
But
assisted
it has become a
reproduction.
very major
So I think
commercial
there's a
enterprise,
very interesting
especially in
future both
clinics
on the clinical
that
and
have
research
an in-house
side.
capacity
And I think outcomes
to do
will
genetic
improve
testing.
substantially
I'll talk
in
about
our
that later.
ability to
But it's long
better,
been recognized that
clearly detect
aneuploidy
embryos
is a
that have the
likely
highest
cause of a
level
significant
of
number of
competence to
spontaneous
continue
abortions.
to
This has
develop
been known for
normally.
quite some time.
Doesn't address the other
And this
factor,
has been
which is really
determined
quite important,
from
and that is the
genetic
uterine receptivity
screening by karyotyping.
phase of,
And this has
of
been the
reproductive
rationale,
of the embryo
for example,
side.
for prenatal genetic
there will be
screening by
remarkable improvements
amniocentesis
that
and
remain
later
to be
chorionic villus
discovered.
sampling.
So with that,
That
I
first
hope you
used
enjoyed this
conventional
presentation,
cytogenetics and
informative,
karyotyping with what is
and
called
I'll,
G-banding,
leave
where
you
you can
with the
actually
future
see the
prospects if you
differential
wanna look at
regions
that.
of the chromosome.
Thank you very much.
Of metaphase chromosomes to detect aneuploidy, translocations, and relatively large deletions by light microscopy. And this method was first used to examine meiotic metaphase chromosomes in residual IVF and gift oocytes in the late 1980s and early 1990s. And in this case, what happened is that gift, which was done quite often, quite successfully, for gamete intrafollicular tube transfer, which was actually more successful than IVF, because at these early days, IVF success rates of 10% were not uncommon, and in some programs 20 and 30% were considered quite good. Now, of course, it's much better. And the question is, what is going on here? Because when you do these transfers, pregnancy rates were pretty low, and this led, in fact, to a use of large numbers of embryos to transfer. In fact, all of the embryos that were produced, usually the cleavage stage is on day 3. And this resulted in unfortunate outcomes for patients, maybe not for the clinic, but for the patients, and that is of higher order gestations that were quintuplets and quadruplets, triplets, etc., that were quite problematic, as you might imagine. So the question is, why put all these embryos back? Because in fact, when you did lower numbers of transfers, let's say 1 or 2, the pregnancy rates were also commensurately pretty low. And so what was going on here? That was the problem. And one of the thoughts was that the genetic issues that we had seen with spontaneous abortions that led to prenatal testing was the same as occurring in IVF, and maybe exaggerated or enhanced by the fact that whereas in natural conceptions a single egg is ovulated, in the case of IVF we produce large numbers using pretty aggressive endocrinology to produce large numbers of follicles to get large numbers of eggs to increase the likelihood of a pregnancy by putting more embryos back. And that has an unfortunate outcome of having higher order gestations in some cases. Now, clinics may have liked that at the time. Some of them did because it was great publicity to have triplets or quadruplets reported. But for the patient, this was not a good outcome. And it was thought perhaps that there was a so-called endocrine storm that occurred that from these types of stimulations that sort of affected the intrafollicular biology of the ovary, where the eggs were growing and developing and ovulating from, that was responsible for the boost in aneuploidies and other abnormalities that were detected. So to detect this, you needed to look at oocytes, and so a lot of gift programs, ours included, did not want IVF after the gift procedure, and so there were a lot of spare and residual oocytes to examine. And in other cases where freezing results were so poor at the time for embryos, just by program freezing, and just were not really terrific, that patients decided not to have their oocytes fertilized, and just those that were selected to fertilize would be put back. And so this was a source of material to look at frequencies of aneuploidy and other types of defects, if you could detect them, in the oocytes. And there were quite a few reports that showed in the 80s that nearly 50% or greater of the oocytes that were examined were aneuploid, monosomies or trisomies, or both. And this was an astounding finding, that in fact a lot of the genetic abnormalities that resulted in non-viable embryos to implantation or to continue or to abort later in pregnancy pre-existed in the oocyte and may be higher than occurs in natural ovulations because of the procedures that were used to generate large numbers of eggs. This was followed by FISH methodology, which is fluorescence in situ hybridization, that used specific DNA probes coupled with fluorophores that was introduced for numerical chromosomal analysis of single blastomeres, physically extracted typically from an 8-cell embryo. And in these cases, the FISH analysis was really quite good because you could actually look at individual chromosomes to chromosome or later chromosomes to determine ploidy and for specific, as it was early used, monogenic diseases such as cystic fibrosis. And this was an important finding, an important advance in embryology detection for genetic normality. And FISH, of course, was not developed for embryos, but was developed for other purposes, but was applied, like in many other things that we'll see, to embryos in general. Ploidy findings were assumed to be representative of chromosomal status of the embryo for purposes of transfer or discard. So when you take a single blastomere out and you look at it and say, well, it looks normal, the rest of the embryo might be normal, or in the 8-cell stage, if it's abnormal, perhaps other cells in this embryo are genetically abnormal and that embryo should not be transferred. And there were also concerns expressed about damage to developmental competence by removing one or more blastomeres at this very early stage, and this was a real concern that occurred. But if we look at how things were done, I just wanted to show you a little bit of history for those who may not be familiar with it. Originally, when we looked at oocyte chromosomes, or even karyotypes from a single blastomere, you needed a metaphase spread or metaphase state. For oocytes, it's a bit difficult because meiotic chromosomes at this stage tended to be more condensed and difficult to image. And in fact, if you look at karyotypes from metaphase in a blastomere, then it was a bit better. And you get something that looks like this shown here for each chromosome set. Now, what was interesting is that early on, this was very tedious because you had to actually take a photograph of a decent metaphase spread. Hopefully you could see all the chromosomes, and you'd have to cut them out individually from a photograph and then align the pairs of chromosomes. And in fact, you may be able to see it here where you have the actual portions of the photograph that's still there and align them. And it's very tedious to do this and to do it right. And not only that, but a lot of the times the chromosome spreads were not that definitive. What changed things, and this came from amniocentesis primarily, was instrumentation that allowed you to look at a slide. The instrument took pictures of the slide and then electronically organize the chromosomes in the right pairs. So you really could use a cursor to move chromosomes around and to align them properly, and that made things a lot easier to do. And so by using this, we were able to look at meiotic chromosomes pretty readily from a large number of eggs and see what the status was. And by doing that, we could see that, for example, here in one of the studies that we published in this case 92 and an earlier one in 88, that we could look at specific groups of chromosomes from oocytes and look at what might be expected to occur from a spontaneous abortion versus what we were seeing in oocytes. And what was interesting in particular is that the G-type chromosomes, in other words, in that particular group, representing the smaller chromosomes, sex chromosomes and 21, 17, etc., were much higher in their occurrence in the oocytes than expected from spontaneous abortions, which are shown at the top line. And so in many cases, 20 or 25 or almost 50% in some patients of their oocytes were in fact aneuploid at this stage. So this was sort of confirmation of what other people had been seeing at the time, that a high proportion of oocytes were chromosomally abnormal, and those chromosomal abnormalities were transferred to the embryo because they didn't preclude fertilization. So this was sort of the first finding that was really quite interesting of human oocytes, again, from IVF cycles, from stimulated cycles. That was really quite important in understanding the origin of the types of defects that compromise human embryos and led, in fact, to the entire pre-implantation genetic testing enterprise. And this is actually not a surprise in a sense, because I remember when I was in college, there was a OBGYN that gave a talk on very early days of studies of fertilization and studies of pregnancy that claimed that 50 to 70% of the time in nature that sperm and egg would meet would not result in a viable offspring or an offspring at all. And so this was something that was sort of astounding to me at the time and got me really quite interested in this field. And later on, years later, I mean, this type of data essentially backed up the contention that really in vivo, in the human, compared to other animals like mice or rabbits, that not very efficient in terms of success after fertilization, by and large. I mean, some extremes exist, of course, but by and large, on average, that the time sperm and egg meet is really not going to result in a successful outcome. And that, of course, is going to, you'll see, increase with age of the parents. From old school karyotyping, which I just described, of looking at individual chromosomes, to FISH, to detect specific gene sequences on chromosomes, was the next step in this progression. And what this allowed you to do was to have greater specificity in looking at specific genes. In other words, specific gene mutations that occurred on chromosomes. And it was a technique that was actually developed not for IVF, of course, but was developed for genetic studies in cell biology and molecular biology. But it was first applied by Alan Handyside and others to embryos to look and try to distinguish embryos that had carriers of mutations such as cystic fibrosis and later others, to avoid the transfer of those embryos. So it was a very useful technique, and this just shows you how it was done. The big advance, of course, came with what's called multicolor FISH, or MFISH, and that detects each chromosome at a specific numerical defect, such as Turner's and mosaicisms and others could be seen. And this was done by having specific probes for each chromosome. So you have a spectral analysis that required a number of different types of filters for fluorescent microscopy, but each chromosome had its specific color, and you could effectively do the same type of karyotype analysis with from a single blastomere and looking at all the chromosomes like you would have done before, but much easier in this respect. And there were some other types of adaptations that allowed you to look at some banding patterns, but they really weren't as distinct for looking at types of things like missing regions of DNA that you could see with what was called G banding in conventional karyotypes. But this was a big advance, because here you could see an example where you have a XO situation, which shown here is at the end is that there's just one X chromosome and no Y chromosome. So in this case, this became the sort of method of choice for doing karyotype analysis from a single blastosphere, and it really produced pretty good results, as a matter of fact, for aneuploidy. But the same condition exists. Does it represent the entire embryo, because you're looking at a single cell? From this M-fish, the next step was to do comparative genomic hybridization. Again, a technique that was not developed for IVF, but became a very important tool in molecular biological and genetic research studies of a variety of applications, both clinical and in research. But competitive genomic hybridization is used to quickly scan an entire genome for chromosomal ploidy and genetic imbalances such as deletions and mutation, and does not need cells that are undergoing cell division. In other words, they don't have to be in a particular stage of the cell cycle like metaphase. And the primary advantage of CG-Edge is the ability to simultaneously detect aneuploidies, deletions, duplications, and or amplifications of any locus represented in an array, with one assay being equivalent to thousands of FISH analyses. And I have a paper that is in your reference list that I think is really worthwhile looking at because it early description of how this works for use in embryos and elsewhere. And these arrays were commercial arrays that occurred that you could buy. Some people made their own for specific applications and their own needs. But the large genetic companies started producing these arrays in various sizes and formats, some on glass substrates, others on a membrane, and essentially deposited hundreds, if not thousands, of individual genes on these arrays. So each spot was an automatically deposited sequence from a particular region of a chromosome, a particular gene, and it allowed you to look at a huge number of gene products and the relative abundance that occurred in a particular sample, a particular cell. And so in this case, for example, you'd see yellow being a normal level of gene expression. Red would be a level of gene expression that was higher, and DNA that was in a lower level of expression would be green. So if you were looking at specific chromosomes, you could determine whether or not a chromosome was present in abundance, meaning trisomy, or in less amounts, which would mean a monosomy. So these were really very really nice arrays. Not inexpensive to do, for example, and it was all done by machine. So you couldn't do this obviously by yourself, but you'd have a instrument that would take these arrays and do the genomics on it in terms of levels of expression, and you get a printout from these arrays that showed which chromosome, or in cases of research or in specific monogenic types of situations, which gene was present in a mutated form or not. And these arrays, of course, now can be done on specific gene sets, metabolism, for example, mitochondrial proteins, signaling pathways, etc., are available. So this is a very important advance because it was much more specific and much more rapid than doing it by previous methods, including the multicolor FISH. So we go from competitive genomic hybridization, which was offered into IVF clinics, to what we now do mostly, which is next-gen DNA sequencing, which is a huge advance, by the way. And that advance has created a family of prenatal, but also pre-implantation genetic testing. We used to call this PGD, that was pre-implantation genetic diagnosis, but that changed to a more aptly termed pre-implantation genetic testing because we're not doing a diagnosis, we're doing a screening process. A diagnosis, you could say, would be for a specific gene or a specific mutation related to a specific disease that you're testing for. So there are very different forms of it, and some of you may know about this, some may not, but there's PGD-M, which tests for monogenic diseases such as cystic fibrosis, Duchenne's muscular dystrophy, Fragile X syndrome, Huntington's disease, sickle cell anemia, neurofibromatosis, X-linked hemophilia, etc. So this is actually used quite often in those patients where you have a known or suspected familial genetic defect that you want to test for so that an embryo that is affected is not going to be transferred. PGDP is pre-implantation genetic testing for polygenic disorders by estimating the risk of developing adult onset disease that are multifactorial by examining a combination of specific genetic variants. So a lot of diseases, a lot of illnesses that are inherited are not monogenic but multigenic. In other words, they're multifactorial and several genes are involved. And in these cases, you're looking for polygenic disorders. And this is done in some cases, not often, but it can be done in some cases for particular genetic illnesses that may be present or possibly transmitted to a newborn. BGTA is the most common done, and it's a genetic screen. So the most commonly used genetic screen is PGTA, that's what it's termed now, and it's used to detect aneuploidies such as Downs, Klinefelter's, Turner syndrome, and with more recent methods to identify structural DNA defects. And this is actually fairly important, such as microdeletions, duplications, small regions where DNA is duplicated, inverted, and where it occurs on specific chromosomes. One of the best known areas, I'll talk about a little bit, for this type of screening is for males to look for microdeletions in the Y chromosome. And there are many microdeletions now that have been detected of very relatively small base pairs in size that are critical determinants of fertility. And so situations such as azoospermia, low sperm count or abnormal sperm have been mapped to specific deletions on the Y chromosome that can be found by looking at the next-gen sequencing of the Y chromosome by this method. And so it's a very important one where you have questions of whether or not genetic defects, deletions, for example, some translocations can occur, can in fact impact on the fertility of the male and also on the normality of the fetus, baby after birth, with defects that don't compromise sperm production but may be transmitted to the next generation in the male. PGT-SR is again, like PGTA, is incorporated in PGTA for structural anomalies, which include microinversions and deletions and duplications and translocations, and likely soon, I suspect, be able to detect what are called epigenetic modifications, such as methylation and other types of modifications that are normal or can be abnormal that occur on the DNA that are important in developmental outcomes and developmental normality. So I would predict that we'll be able to detect certain epigenetic modifications that are inconsistent with a normal embryo's development and its likely development after transfer. So now, next-gen sequencing is the most used method for pre-implantation genetic testing. And it basically, the methodology is pretty straightforward. It involves parallel sequencing by a machine of multiple DNA small fragments, or small DNA fragments, that are recombined to determine the entire sequence. And the benefit of this, which is really quite profound, is its high throughput significantly increases the speed at which an individual's genome is sequenced and displayed for analytical purposes. And the sequencing is a remarkable change in how we do things considered to how the original genome project, Human Genome Project, was done. I mean, it's very fast. And it's also, for research purposes, pretty inexpensive to do these analyses. And AI, however, artificial intelligence, is a recent, I'll talk about as well, is a recent enhancement that has the potential to markedly increase the genetic information content because it detects and prioritizes genomic variants linked to certain diseases. And it uses this by using large population-derived databases of normal and affected individuals. So by adding machine learning and analysis in the process of looking at these next-gen sequences, you can pick up additional information from a large database that will increase the specificity and utility of the information derived. And this has now been adapted, and as you'll see, for embryo use. But all of this taken together, whatever methodology, confirms what's been known for a long time, that the frequency of chromosomal aneuploidy rises with maternal age. And this is a typical, well-observed phenomena, and this is the type of pattern that you see, that as maternal reproductive age increases after about age 30, so does the incidence of aneuploidy. And this is something that is known and actually can be used to determine whether or not a particular patient should be subjected to pre-implantation genetic testing based on their maternal age and also on family history. So this is very useful information in this regard, and this just confirms what has been known for quite some time, even from spontaneous abortions. One of the problems that has occurred, that is a kind of interesting one, that has occurred early in the analysis of embryos by fish, for example, was mosaicism. And what is mosaicism? And that is that you have some cells that are normal and some cells that are not. So, for example, a healthy embryo would have two chromosome 21s. An unhealthy one would have Down syndrome, for example. It would be detected with 3 chromosome 21s. Or an embryo with Klinefelter syndrome, where you have XXY karyotype. Now the problem is that if you see this in one cell, chromosome 21, of an 8-cell embryo, that embryo would not obviously would not be returned. Healthy embryo, of course, yes. Embryo with Klinefelters, suggestive that in fact maybe an 8-cell embryo that has that would be not replaced or transferred or cryopreserved. But what happens when you have these sort of mosaicisms occurring in a trophoblast biopsy, which is pretty typical done now, where you're dealing with more than one cell at a particular stage, usually cleavage? What do you do with those embryos, and what is known about mosaicism, and how important is it? Because it can be confusing to get a report back with a situation where you have a complex aneuploidy, where some cells may be normal and some cells may be abnormal in the trophectoderm. What do you do with this information? Here, for example, is a typical PGA report from using next-gen sequencing, and it's taken from Taguchi, which is a very good review that I put into your reference list. And what it shows is that, in fact, here's a normal embryo that you can see here in the upper right, in the first panel. The second one being mosaic. This is how it comes out, and you have regions where you have obviously have some cells that are normal, some cells that are abnormal, and then the complete aneuploidy showed in the bottom panel, which is clearly distinguished by the size of the increase in the DNA at one loci. So here it's telling us that you can fairly well see mosaicism, some are more profound than others, compared to the normal. So what do you do with this information? I mean, do you put an embryo like this back? And this has been sort of contentious. And there's been a lot of work done by quite a few people in this regard, and it says that does more detailed genomic information from PGD findings create more confounding issues, such as mosaicism, for transfer, cryopreservation, or to discard the embryo in the decision-making process. Because for the patient, if you get a report back that says, well, you have this mosaicism, but we're not quite sure if it's going to be important or not, what do you expect to do, and how do you have really good informed consent when you're not sure? So because of that, a lot of the work that has been done from quite a few programs now has come together to say the following, that typically most embryos are considered normal if they have under 20% of aneuploid cells at the trophoblast stage, where you have essentially blastocyst. So those embryos will likely be put back, although there's some concern. And you might say to patients who have done this that, well, if you're really concerned, you can have a fetal maternal blood test done to look at the genetic composition of the fetus based on cells that are shed from the placenta. And in maternal circulation, it's really now quite common to capture those cells, and this is representative of the fetus itself, so you can be reassured or not as to the normality of the embryo that was put back. 20 to 40% is considered low level, and that's again where you might recommend to have a subsequent genetic test using maternal blood. 40 to 80% is considered high, and greater than 80% is considered abnormal. And in those cases, the transfer of the embryo is not recommended. So you can use this information from these types of testing for that purpose. So often, the detection in next-gen sequencing reports of structural anomalies, microdeletions, for example, or mosaicism, has been used to err on the side of caution, even at low levels, to preclude transfer or cryopreservation. But some widely discussed published studies and comments in the popular press, and these are quite recent, and some comments from prominent members in the IVF field, suggest that thousands of embryos may have been needlessly discarded because recent outcomes indicate that such embryos can result in normal babies. And this has been discussed at some of the large conferences, IVF conferences, as to what do you do in these cases. And this has created quite a dilemma, okay? And this has created clinical and patient confusion about whether and which PGT findings are truly actionable, put back or not put back. And in some notable instances, at least in my country, patient-initiated litigation, court lawsuits involving IVF programs that failed to transfer embryos when all were suspected based on PGT results, including mosaicisms. So this is a complication of getting a lot more information that can occur. And so it's not all clear-cut, but I think it is clear-cut that it is evident that some mosaicisms and some situations that occur from the reports of genetic testing are not necessarily terminal in terms of decisions about whether to put the embryo back or not. And embryo replacements have been done with normal outcomes, and in some cases with follow-up genetic testing by DNA captured from maternal blood, fetal cells from maternal blood. So there's a lot of information that's obtained, and yet it's not all that drastic in terms of outcome, because positive outcomes have been reported, a lot of them. So with the ability to do a subsequent follow-up genetic test that's not invasive, like amniocentesis or chorionic villus sampling, has made the decision process and informed consent a little bit more easier to obtain. So, but now is the big question. And the big question is any of this really necessary? And PGT or any of these genetic testing is really necessary as a screening process in all cases. And indeed, the accepted value of PGT-A has recently come into question. And one of the really good studies, which I recommend you reading, published in the New England Journal of Medicine by Yan et al. And they report that live birth with or without pre-implantation genetic testing for aneuploidy. Okay? So here is one of several studies that have questioned the use of this as a general screening system in all cases. So for trials evaluating all IVF patients, rather than restricting it to certain ones that are at risk, for trials the central question is, in trials evaluating PGT-A, the cumulative live birth rate reflects the possible effects of the practice of discarding some embryos that might have led to a live birth if they had been implanted. Such data cannot be captured, they claim, when only the first embryo transfer is analyzed. And some programs are reluctant to do frozen embryo transfers on these embryos to begin with, based on first pregnancy outcomes. So what was this study design? It compared cumulative live birth rates after PGTA based on a combination of morphological criteria and next generation sequencing on the same embryos with the rates after conventional IVF based on morphological criteria alone. And there's a lot of data on morphology that is used, for example, in algorithms from time-lapse machines, culture systems like the embryoscope, that uses those algorithms based on what embryos are doing and when, certain landmarks of development that occur, and that they occur at the right time, through the blastocyst stage for the selection of embryos. So there's nothing else but those algorithms and embryo appearances and morphokinetics and morphodynamics and time that the embryoscope picks up. And that's been used for selection, which has had actually very good results at the blastocyst stage. However, in the PGA group, a euploid blastocyst was chosen for transfer. That is, only the ones that were examined by genetic analysis. In the conventional IVF group, a blastocyst was chosen based on morphology and morphokinetic criteria alone. If a live birth was not achieved after the initial transfer and there were remaining euploid embryos in the PGA group, or morphologically transferable embryos in the IVF-only group, subsequent single-cell embryo transfers were performed. Now, what they concluded, there's a large number of embryos, by the way, in patients that were done in the study, and their conclusion was, among women with 3 or more morphologically good quality blastocysts, conventional IVF resulted in a cumulative live birth rate that was non-inferior to the rate obtained with PGTA testing by next generation sequencing. They were similar. So their argument was that for general purposes, if you're going to have a mandatory program where everybody gets genetically screened for embryos, is not worth it. And you could argue that in this case you might reserve the use of this type of genetic testing for those patients that are at risk because of age, previous fertility history, previous birth histories, or familial related problems that could be transmitted to the next generation. So this was actually quite useful information, and it's always discussed at large meetings as to whether or not the entire enterprise that has been driven a lot by commercial companies and the ability to do this, and sort of the imperative to make sure you're not going to be in a situation where you're going to be knowingly put back an embryo that had some obvious faults on the genetic testing and have a bad outcome, and from that bad outcome be liable for damages, etc. So a lot of that plays into the decision-making process, at least in this country, in my country. So the introduction of non-invasive genetic testing, that's the next step. And this is an interesting one, because in this case, what you want to be able to do is to get material for analysis without actually having to do something invasive, like trophoblast biopsy. Now, the morphological testing from PGD, from embryoscopes, which is based on morphokinetics and morphology and time, and specific landmark events known to be important, is essentially non-invasive, except all of those procedures require ICSI, because you're looking from the very earliest stages of development, from the pronuclear side of things, when pronuclear formation occurs, when pronuclear dissolution occurs, when the first cell division occurs, etc. That these are important criteria for morphological selection going forward to the blastocyst stage. So ICSI is done in these situations, so it's not necessarily non-invasive, but it's invasive for the method of fertilization. So the introduction of non-invasive pre-implantation genetic testing is a fairly new aspect of this, and may be an important breakthrough for validating embryo chromosomal status, or is it more of the same as related to actionable information? In other words, we have the same problems of mosaicism and microdeletions and getting complex patterns of genetic testing back where you have complex aneuploidies. Some chromosomes are present or monosomic. Some chromosomes are trisomic, and you have these deletions that are occurring on various levels in different chromosomes. So what do you do with this information? Okay, is it more of the same? So these are the current non-invasive methods that have been introduced. One is the analysis of static and time-lapse images during culture, with or without artificial intelligence enhancement. In other words, using data sets that add to the information content obtained from the analysis. Those static images is having embryologists look at embryos at specific times when specific events that are thought to be important, nuclear breakdown, first cell division, when compaction occurs, the nature of compaction, etc., first blastocyst formation. That's a lot of work to ask. So those are static versus the time-lapse, which does it for you. And of course, in the case of static observations by embryologists, you're not having the addition of AI algorithms that add databases from large collections of embryo observations to the decision-making process. The other ones, which are really quite interesting, is collection of DNA from spent culture media after about 5 to 7 days of culture. And the question is, why is DNA in culture medium? And I'll get to that in a moment. And the other one that has been introduced, which is the manual collection of blastocelic fluid, looking for DNA in that fluid. And that DNA is obviously going to come from either inner cell mass or come from the trophectoderm. And this has been actually done quite interestingly enough. So let's talk about the introduction of artificial intelligence. So the introduction of artificial intelligence algorithms in non-invasive embryo selection. And this is, again, done in systems like the EmbryoScope, which is doing time-lapse, but you have the addition of information coming from previous cycles that has been used to support whether specific landmarks, developmental events, are in fact the important ones or not. So that's what the AI is telling us, telling those algorithms. So the question is, can the inclusion of artificial intelligence algorithms in PGA significantly increase the accuracy of aneuploid euploid identification, presence of structural defects unknown, to better select embryos suitable for transfer at the blastocyst stage? So you have both the artificial intelligence from NGS sequences that are done to add to the NGS analysis, and in cases of morphology, from morphological results related to outcomes that are considered to be relevant and important in the analysis of which embryo to select, either from time-lapse or to select for after DNA sequencing. So this is currently done. It's quite a bit of information about this available. So for this to better select embryos based on the addition of AI information, you need evidence-based outcomes to prove a significant increase in ongoing pregnancy rates occur and live birth rates occur compared to static morphology, or which is the EmbryoScope or the next generation sequencing alone, or in combination. Okay, but no one usually is going to. If you're going to do one, you're not going to do really the other. So it's really not like the studies I just mentioned, but it's really either morphology or it's going to be DNA sequencing. So can this add a lot of information that's useful? And that, if it does, it's actually quite important. So I'm going to talk about two studies that I'd like you to look at. I think they're very important, and they just came out this year. So two recent and detailed reports suggest that the use of their described AI platforms and algorithms has the potential to achieve these goals, which I just mentioned, by outperforming embryologist grading at the blastocyst stage. And one study by Barnes, which was retrospective, with over 10,000 blastocysts used maternal age, morphokinetic, and morphological characteristics typically applied for blastocyst grading. I think many of us who in this field know about those criteria. And in this instance, static images obtained at 110 hours post-ICSI. And these non-invasive findings were correlated with corresponding PGTA results. And what they found was that, in fact, the morphology and what they found by the imaging at 110 hours post-ICSI correlated pretty well, about 75% or more, for aneuploidy, euploidy, and complex aneuploidy determinations. Not 100%, even in the case of next generation sequencing, you can have samples that provide no signal. So it's not 100% in those cases. You can't get a signal from the embryo, so you don't really know. But greater than 75% is pretty good, and it'll probably get better. And these early findings show promise for ploidy and may especially be useful if ICM, intracellular mass characteristics associated with developmental competence and normality, can be introduced in the AI algorithms with or without time lapse. So static images by an observer or by machine related assessments. So now you have both trophoblast and you have intracellular mass characteristics, if you can define better the competence of the embryo. So I suspect that 75% value will increase significantly going forward. Another highly recommended study, and I think it's an excellent review by Yang and Borman, again in this year, discusses the current state of artificial intelligence used in embryo ploidy grading and its limitations. And here, for the first time, we're really talking about limitations, including issues of mosaicism, structural defects, which include, as I've mentioned, microdeletions, inversions, etc., and use of image-based analysis, and more importantly, the source of the DNA. Where is it coming from? Is it coming from culture media? Is it coming from blastocytic fluid? Is it coming from biopsies of trophectoderm? And their conclusion is that even with possible confounding factors, AI-based ploidy prediction has the potential to improve pregnancy rates and reduce the cost of IVF cycles. And this is important, by the way, because here what we're seeing is that their conclusion, based on, again, a large number of patients, says it has an ability to add AI to reduce the cost of the cycle and the accuracy of the reports that come out in the decision-making process of which embryo should go back and which embryo likely should not go back. But I just want to put a note in, because this is from what I've seen at going to various conferences where this has been discussed, and of course before 2023, when a lot of this information and methodology was discussed as coming into its fore. Not all are this sanguine about artificial intelligence and image-based analysis, owing to the question of whether undetected mosaicisms and structural defects exist, or even when they're detected, may compromise actionable decisions for transfer. This is especially true if the affected embryos can result in normal outcomes. You just don't know. And this would tend to argue for a gold standard finding, and I think the gold standard finding will remain the genetic screening, which looks at the entire genome, separate from morphology and separate from other types of analyses. So this is going to be important. And again, if that is true, then the current considerations for artificial intelligence for non-invasive ploidy detection are cost versus benefit. And likely needed, for example, ICSI in these cases. You can't really do, if you're going to start from the very beginning, unless you have to denude cells shortly within a couple hours after fertilization, and then put them into a time-lapse system or start your inspections at very early stages. So maybe the likely need for ICSI, although I think in conventional IVF can work as well, but it is more time-consuming and operator-consuming and probably costly. Likely need for multiple time-lapse culture systems to accommodate embryo numbers to be examined, especially in an active IVF program. Meaning that if you're going to use AI in a machine learning situation built into a time-lapse embryo culture system, you're going to need a lot of them to do. Or you're going to just have to reserve use for patients at known risks, age or family history, as I mentioned before. And there's also the cost of non-public AI algorithms that's not included in the culture system. So many include, there are some that, in the studies that I've referred to in the previous slide, do refer to their public domain AI systems, but others are going to be proprietary and come only with the culture system that you're using, if it's a automated machine like the EmbryoScope, for example. And then the issues related to the detection of mosaicism, structural defects, which I've mentioned before, including translocations, and which chromosomes, if trisomy or monosomy, are affected. I mean, how do you know which ones are from either static inspections by an embryologist or from the machine? And it's not clear that information will be likely. But you'll likely need an IVF program to self-validate with coincident genetic screening, genomic screening at the blastocyst stage. So you can start it, but you probably will need self-validation to make sure that the system is showing you what is expected. And one solution that's been mentioned, I think it's a reasonable one, is to reserve these types of advanced methodologies, not using just genetic screening alone, for couples considered at risk or where PGTA would be recommended. However, there is another non-invasive method of PGTA that has recently been introduced, and that is one that uses cell-free DNA for next-generation sequencing. So, non-invasive PGT using cell-free DNA. And this is actually an interesting aspect of this whole enterprise of genetic testing. Prenatal genetic testing has evolved from invasive amniocentesis, CVS, to cell-free capture of DNA in maternal circulation, shed by apoptotic placental cells. So this is now sort of used routinely for genetic testing, rather than going through amniocentesis, unless you have to look at specific things, or chorionic villus sampling, which requires cell culture, as does amniocentesis, and is much more expensive. Similarly, DNA detected in spent culture medium is by definition non-invasive, whereas blastocelic fluid, which has also been studied, obtained from manually collapsed embryos, is considered minimally invasive for PGTA purposes. In validation studies, some reports describe an acceptable level of concordance, meaning it's pretty good, with DNA extracted from corresponding trophectodermal biopsies, while other reports are less so optimistic. But in general, it's getting to the point where the concordance is not that bad, because you're looking at DNA extracted from, or expressed by, probably released from the trophoblast, trophectoderm. When or how, why or how DNA occurs in culture medium or blastocelic fluid is unclear, but it is clear and known that apoptosis occurs in the trophectoderm of several species. For example, in mice, and certainly in bovine, and certainly in species like rabbit, it's very, very pronounced and very normal that it occurs. So it's likely that at the expanded blastocyst stage in human, it has a similar origin for this DNA, especially in blastocelic fluid. But embryo DNA derived from culture medium is truly non-invasive, and it seems from several studies that, as I said, concordance with trophoblastic biopsies is superior to that of DNA from blastocelic fluid. However, so you may want to use the culture media and not undergo a manipulation to manually express blastocelic fluid. However, for both sources, culture media or blastocelic fluid, significant PCR amplification is required for meaningful next-generation signals. So many of the same issues of concern with nuclear DNA exist. that occur with intact cells may apply, and this includes allelic dropout, levels of amplification, fidelity of amplification, etc. For culture medium, however, unlike blastocytic fluid, robust denudation of cumulus and coronal cells is a must to prevent maternal contamination for even from a handful of residual cumulus cells or coronal granulosa cells, and this may require aggressive ICSI. As with conventional IVF, it can be difficult to assure complete maternal cell elimination. So, what are the relative benefits of this? The relative benefit of cell-free DNA for non-invasive PGA testings are worthwhile, remains to be settled, but is conceptually relevant in IVF. In some reports, blastocytic fluid, as I mentioned, has been considered unacceptable for PGTA owing to high levels of amplification failure and or high levels of discordant rates with coincident biopsy trophectoderm extracted DNA. And collapsing the blastocele prior to vitrification has been suggested to improve post-thaw viability, increase the permeability of the cryoprotectants prior to vitrification, and has been suggested, as I said, to improve viability after thawing, as the blastocyst cavity should reaccumulate fluid. In culture, blastocysts undergo periodic collapse and transfer, for example, and re-expansion. So this is not a toxic event. However, it might be interesting that full re-expansion is not necessarily given, and failure to expand or return to a pre-collapse state, which can occur. It can occur when you look at blastocysts in culture, that they undergo this contraction, expansion, contraction, expansion, and we've seen, and others have seen, that you get a contraction and the blastocytic fluid is out and it doesn't re-expand, or it starts to re-expand and stops. So, however, full re-expansion is not a given, and failure to expand or return to a pre-collapse state when you do these extractions of blastocytic fluid may indicate a reduced metabolic capacity on that embryo, which in itself could serve as a further marker of developmental competence. So it remains to be determined whether or not these methodologies are useful, comparing blastocytic fluid to culture media. In fact, I would suggest that if the culture media effects are real and the amplification process problems are potential artifacts are addressed, that this may be a very important method in the future for doing next-gen sequencing without worrying about developing the embryo in a machine that is going to do morphological assessments. Although additional validation studies are needed, it would seem that currently collecting cell-free embryonic DNA in spent culture media could be the most likely cost-effective means for PGTA, allowing the power of next-generation sequencing to determine ploidy structural defects, translocations, and for PGTM, monogenetic mutations. So what is the take-home message from this talk? And many aneuploid embryos will self-deselect by arresting development prior to the expanded blastocyst stage, even with extended culture. What this means is that a lot of embryos we know don't go to blastocyst, and those will self-select, and those are not going to be transferred or cryopreserved. So you're really left with a portion of the embryos that you start out with that go to a stage where you really have to be concerned about processes of mosaicism and aneuploidies, etc., certain aneuploidies. At present, I would say, if you believe genomic sequencing is the gold standard, and this is individual determination, to detect genetic issues that may compromise outcome, even morphologically stage appropriate for ICM and trophectoderm, then DNA sequencing is indicated. In this regard, DNA in spent culture media has a promise of avoiding invasive intervention and should be cost-effective for the patient, and if proven to be equally accurate as cellular DNA for NGS, an alternative may exist to time-lapse imaging even with artificial intelligence. And I want to end by talking a little bit about the future prospects. Modern cell biology has advanced tremendously in its ability to examine molecular, metabolic, and specific gene expression at the single cell level. often simultaneously in the living state. This is a remarkable developments in this field of what you can see. RNA-seq, DNA-seq, all sorts of things that you can do, epigenetics, at the single cell level. Combined with various omic platforms, metabolomics for example, insights into normal and abnormal cell functions can be assessed, and if applicable for cells extracted from late-stage pre-implantation human embryos, similar insights into post-transfer normality might be estimated as a final screen for developmental competence. While combining PGT and these types of embryo studies may be asking a lot, because you're doing a number of different tests at the same time, one lesson learned from over 4 decades of clinical IVF is that the imperative to improve outcomes relies both on skilled embryologists and the continual development, testing, and application of viable new technologies that can be clearly provide different streams of developmentally relevant information. In this presentation, the evolution of PGD from chromosomal karyotypes to NGS with the inclusion of artificial intelligence is a clear example of a progression that serves this imperative. And I think we can really anticipate that this will continue to evolve and contribute to one of the most important and successful areas of medicine, and that is assisted reproduction. So there are a lot of things that I think will be coming, including epigenetics and including other types of DNA analyses, that will be really, really quite important in assessing developmental competence and useful in selection, whether they can be streamlined not to require special instrumentation, but can be done relatively inexpensively and relatively rapidly, as is the case with DNA sequencing. As DNA sequencing has evolved, remains to be determined. But I think the future is really, really interesting for both the clinical and the research side in this. And I think what will happen is that the increased use of these instrumentations and technologies will really increase markedly outcome in IVF for the patients. So with that, I'll leave you, and I hope you find this